Hello, good morning, students. Today we will study from the biomolecules proteins. Next type of biomolecule is a protein. So, what are proteins? The proteins are fundamental structural materials of animal bodies. Proteins in the form of enzyme play important role, prime role in all physiological reactions processes. The name protein derived from the Greek word proteos. The meaning of proteos is a primary or prime importance. So the compound having prime importance, these are the proteins. The nutritional source of protein are milk, pulses, nuts, fish, and meat, where we get naturally protein. In which food material? These are milk, pulses, nuts, fish, and meat. Chemically, proteins are polyamides. So they contain poly means many and amide linkage means C double bond O NH linkage. So these are the polymers and therefore they have high molecular weight. The monomers present in protein, these are called as amino acids. Now we will study what are amino acids, what is their general formula, and examples of amino acids and classification of amino acids. So these alpha amino acids, these are obtained when protein undergo hydrolysis. Protein on hydrolysis give alpha amino acids. This alpha amino acid contain, this is a carboxylic acid having alpha carbon with amino group, that is NH2 group. Therefore they are called as alpha amino acids. The general formula of alpha amino acid is RCH NH2COOH. General formula is RCH NH2COOH. So in this table it is given that the different examples of natural alpha amino acids, and here the asterisk is marked. These are essential alpha amino acids. So these amino acids are classified into three types. One is a neutral, second is acidic, and third is a basic. So in neutral alpha amino acid, the NH2 group and COOH group, they are equal in number. If number of NH2 groups and number of COOH groups are equal, then it is called as a neutral alpha amino acid. If alpha amino acid contains more COOH group, that is a carboxylic acid groups, than NH2 group, then these alpha amino acids are called as acidic alpha amino acids. Then basic alpha amino acids contains more number of amino group, that is NH2 group, than carboxylic acid groups. These are called as a basic alpha amino acids. Now we will study this type of amino acid, then name of amino acid, the R group present in amino acid. We know general formula RCH NH2COOH. So this central carbon is bonded with this one R group. second is hydrogen, third is NH2, and fourth is COOH group. So all these amino acids have this hydrogen, NH2 and COOH. These are the fixed groups bonded to central carbon. Only difference is R group. So R group is different in different amino acids. Then from the name, the 3-letter symbol and 1-letter symbol is given for these amino acids. We will study one by one. So first example under neutral alpha amino acids is a glycine. This glycine have R group is hydrogen. It is a very simple example of alpha amino acid. Here the 3-letter means first 3 letters from this glycine, it is given as a 3-letter symbol GLY, and 1-letter symbol is first letter, that is a G, stands for glycine. Then second example is alanine. Here R group is CH3. 3-letter symbol is first 3 letters ALA, and 1-letter symbol is given as a capital A. Third example is valine. Here asterisk is given, therefore it is essential amino acid. Here R group is Me2, means CH3 2 times CH. It is a isopropyl group, and the 3-letter symbol given as the first 3 letters from the name VAL, and symbol is V. Fourth is again essential amino acid. It is a leucine, and R group is Me2, means CH3 2 times CH2. Here 3-letter symbol is LEU, and 1-letter symbol is capital L. Fifth example is isoleucine. It is again essential amino acid. R group in isoleucine is CH3, CH2, CH. Me means CH3 group. This is R group. Here the 3-letter symbol given is IE. Here I from iso, ILE, sorry. It is I from iso, L, it is a small letter from the leucine, and E is the next letter to L. So ILE, and the 1-letter symbol is I. Next example is asparagine. R group is NH2COCH2, symbol is ASN, and 1-letter symbol is capital N. Seventh example is glutamine. Here R group is NH2COCH2CH2, symbol is GIN, and 1-letter symbol is Q. Next is serine. R group is OH, CH2, symbol is SER, and capital S. Next is threonine. R group is CH3, CHOH, symbol is THR, and 1-letter symbol is capital T. This threonine is again essential alpha amino acid. Next example is cysteine. Here R group is SHCH2, symbol is CYS, and capital C. Next example is methionine. It is again essential amino acid. R group is Me, means methyl, CH3, S, CH2, CH2. Here symbol is given as MET and capital M. Next example is phenylalanine. It is again essential amino acid. R group is PH, CH2, symbol is PHE, and 1-letter symbol is F. Then next example is tyrosine. Here R group is para OH group, that is C6H4 is a benzene ring bonded with para position OH group and CH2 group. These are para position to each other. Symbol is given as a TYR and one letter symbol is Y. The next is tryptophan. It is again essential amino acid. R group is benzene ring bonded with 5-membered heterocyclic ring with nitrogen, that is NH group and CH2 group. The symbol is TRP and one letter symbol is W. The last example in neutral amino acid is a proline. Here R group is 5-membered ring with NH and COH group. This is the entire structure actually for proline, not R group only. Here it contains COH and NH group with 5-membered ring. The symbol is given as a PRO and one letter symbol is capital P. So these are the examples of neutral amino acids. Now we will study acidic amino acids. Only 2 examples are there. One is aspartic acid. It contains R group as a COH, CH2. Symbol given is ASP and one letter symbol given as a D. Then second example of acidic alpha amino acid is a glutamic acid. Here R group is COOH, CH2, CH2. Symbol given is GLU and one letter symbol given as a G. Then the next type of amino acid is a basic amino acid. They contain more NH2 group, more amine group than the carboxylic acid groups. Here 3 examples are given: lysine, arginine and histidine. All these are essential amino acids. R group NH2 CH2 4 times. Then in arginine NH double bond C NH2 NH CH2, and in histidine this 5-membered ring with 2 nitrogen, two double bond and CH2 group. Here the symbols are LYS, ARG and HIS. One letter symbol is K, R and H. So this is classification of natural alpha amino acids. There are total 20 alpha amino acids. They are classified into neutral alpha amino acid, acidic alpha amino acid and basic alpha amino acids. We had already studied the general formula of alpha amino acids R, CH, COH and NH2 group. This is alpha carbon is a chiral carbon because 4 different groups are bonded. Here the Fischer projection formula can be drawn. So this NH2, CH, COH and R. Here it is L-alpha amino acid, and in glycine only this central carbon is not asymmetric, that is achiral. That is only one example. That is the first example. It is a glycine in which this central carbon is not chiral, it is achiral because 2 groups are the same on the central carbon, that is hydrogen. Then if alpha carbon atom in alpha amino acids is obtained by hydrolysis of protein has a L configuration. The L-alpha amino acids are represented by Fischer projection formula. Here the central structure is shown. This is a Fischer projection formula and this is having L configuration. The symbol R in the structure of alpha amino acids represents the side chain and may contain the additional groups. If R contains the carboxyl group, amino acid is acidic. If R contains primary, secondary or tertiary amino group, then they are basic. The other amino acids having the neutral or no functional group in the R group, they are called as a neutral amino acids. The alpha amino acids have trivial names, means common names, generally represented with 3-letter symbols or sometimes by 1-letter symbol. This we had already studied in this table, 3-letter symbol and 1-letter symbol. Then 10 alpha amino acids from this list cannot be synthesized in human body. The amino acids which cannot be synthesized in human body, these are called as essential amino acids and they are obtained through diet. These are called essential amino acids and they are marked with asterisk in the table. Then alpha amino acids have high melting point. They are water soluble crystalline solids, unlike simple amines or carboxylic acids. Simple amines and simple carboxylic acids, they do not have this type of physical properties like high melting point, soluble in water. So these are the properties of alpha amino acids. These properties are due to peculiar structure called as a Zwitter ion structure. So we will study now Zwitter ion structure of alpha amino acid. So alpha amino acid molecule contains both acidic means COH group, carboxylic group, and the basic group as a NH2 group. The proton transfer take place from the COH group, that is acidic group, to amino group and forms a salt which is dipolar ion and it is called as a Zwitter ion. We will study in this structure how Zwitter ion is formed. So this structure is the Zwitter ion containing one positive and one negative charge. Amino acid can exist in different forms depending upon pH of aqueous solution in which they are dissolved. Consider this example of Zwitter ion and the other forms of alanine. We know alanine contains CH3 group. First, the general formula of alpha amino acid is R, CH, NH2 and COH group. So this lone pair is present on nitrogen in amino group and the carboxyl group contains the proton that can be donated to this amine group. So if proton transfer take place, this structure is Fischer projection formula and therefore central carbon is not shown. Here the NH2 group becomes NH3 group because one proton from this carboxylic group that transfer take place and that gives the NH3 plus group, that is called as a ammonium group. So here remaining groups are HR and COO minus, and this ion containing positive as well as negative ion, this is called as a Zwitter ion. This is a general formula for the Zwitter ion of alpha amino acid. Now we will take a particular example, alanine. Here alanine contains R group as a CH3. So the groups are here overall positive charge, therefore pH is less than or equal to 2, that is acidic pH. Here this NH2 group, here there is a mistake, actually it must have the NH2 group, then CH, CH3 and COH. Then one proton transfer take place that gives NH3 plus and COO minus. Remaining groups are hydrogen and CH3. This is a Zwitter ion of alanine, no net charge, and therefore pH is nearly equal to 6. Then this is negatively charged, that is, if proton is lost, then it contains -1 overall charge and pH is greater than or equal to 10. So these are the three forms of alanine. Then we will study peptide bond and protein in the next lecture. Thank you.